Welcome to the latest instalment in the Fungisphere webinar series. My name's Associate Professor Justin Beardsley, and today I'm coming to you from the beautiful Camperdown campus at the University of Sydney. With this series of talks, we're tackling all things mycological, and that ranges from pathogenic fungi right through to the productive uses of these fascinating organisms. Throughout the series, we feature researchers based here at the University of Sydney, but also from around the world, coming together to share their research and their insights. Today, it's my pleasure to welcome Professor Fran Lopez-Ruiz from Curtin University. Fran focuses on pathogenic fungi of plants and how they evolve resistance to agricultural antifungals. He's going to talk about some really interesting genomic approaches he uses in surveillance of the emergence of resistance, and he's also going to make a strong case for why we need to use One Health approaches to tackle this emerging global problem. So let's hear from Fran.
Hi everybody. My name is Fran. I work at Curtin University on fungicide resistance and in basically agriculture, but my background is clinics. Been working in hospitals for a while, especially in involving the HLA system and in transplantation. But basically, then I did a move to plants. They complain less, I imagine, and that's basically what I'm doing at the moment. So I've been leading a group or working on fungicide resistance for the last 10 years. And so I'm going to be basically giving a little bit of an introduction to the problem, just trying to establish some parallelisms between the problem in clinics and agriculture very briefly, and then I'm just going to target two examples that are just driving agriculture crazy at the moment. How do I...
Okay, so we'll just...
Sorry, I'm a caveman.
Just like this. So...
Excellent. Okay, so I guess that the main question is, why do we have a problem with fungicides and fungicide resistance? And I imagine that if we try to establish a relationship between what happens in the field and clinics, it's actually very easy. Microphone better here. Very easy to understand. So just to try to draw that picture. So just try to imagine that everybody decided to start taking antibiotics prophylactically. The same ones always, not having an alternation between different type of chemicals, using them to treat, diseases that are actually not affected by antibiotics, like for example, viruses, right? Taking them at a high dose, that's a super low dose, right? And exposing the entire population. That's what's happening pretty much every day in agriculture. So we're using this sometimes, very high doses of fungicides in homogeneous populations that normally are bred to produce more yield and not to withstand a disease. So, and the sheer size of the problem is just staggering. So there's a massive amount of chemicals that go out into, agriculture the whole time, and this happens, repeatedly, as we know. So resistance develops in the same way as it, for example, develops, in clinics, animal husbandry, animal production. So we use a fungicide, right? And this fungicide selects for individuals that are resistant. In this graph here, we can see the resistant individuals as being the black dots, right? Sensitive individuals. I don't know if they can see my pointer. Can they see my pointer? Probably not. Okay, all right. So black dots, resistant individuals, blue dots, sensitive individuals. And that basically produces selection. So then we come back with another application with a chemical, right? And the selection basically strengthens. And, over time, that leads to an accumulation of the resistant individuals. Just to provide some perspective on the problem. So fungicide sales just keep going up. This is science stats, already a bit old from Australia, but you can imagine that, this trend just keeps going up, right? This trend is the same in every developed country, okay? Now, the number of registered chemicals or registered fungicides keeps going up, but this doesn't mean that we've got more molecules available. Actually, the number of chemistries available decreases, and that's why we've got that 0.7 number in there. So what increases is the number of brands. And because, they're quite complex, sometimes, growers get confused. So this number keeps decreasing, not only because of resistance, but because of deregulation. So most of the big market for Australia is Europe, and whatever happens in Europe in terms of regulation with chemicals ends up happening here as well. The number of cases, fungicide-resistant cases in the field, this is not in the lab, actual field consequences, so it's over 400 around the world. And in the last few years, the majority of the cases are actually coming from Australia. So just to give you another interesting number, the biggest fungicide resistance outbreak ever recorded in the world was in Western Australia a few years ago. So it ended up costing growers more than $300 million over 3 years. So what's the situation at the moment? So when we started working on this, it was only one case of fungicide resistance that we reported. That was more than 10 years ago. And now fungicide resistance, many sorts, sensitivity, resistance, also laboratory detection. So things that we can see in the lab, but we cannot see in the field just yet. So they are just over the place, right? And this is just for grains. And the cases just basically keep piling up. Just a new case that was detected a few months ago in South Australia. But this is not just grains. So for example, in botrytis, so the situation is actually even worse. Botrytis is a disease that affects many crops. And this is a study that we conducted, one of our master students conducted last year. So we collected samples from different supermarkets, and we're not going to say which ones. And we isolated more than 300 isolates. And what you can see in this figure is actually the frequency of the resistant isolates towards the chemicals that are mainly used for the control of botrytis. As you can see, in many cases, that frequency is virtually 100. Now, even more interesting is when you look at the different modes of action towards these isolates were resistant. You can see that there are multiple cases where these isolates were resistant to more than one mode of action. Actually, even up to 7, all right? As you can see at the right-hand side of the graph. So this is just a reflection of what's the selection pressure on how much of these chemicals are we using, okay? And just another of these bigger figures just to provide an idea of how important fungicides are for agriculture. So in this table, I've got 4 diseases of wheat and barley. And there we've got the potential loss, the actual losses, obviously, they're actually lower because we've got some control methods in place. And then, the share of that actually savings because of, genetic control, cultural control, or fungicide control. So fungicide use is actually comparable to genetic control, to the breeding of resistant varieties, which is very important. So we've got multiple different modes of action. There are many of them, but mainly we use 3 of them, okay? So DMIs, the methylase inhibitors, they're actually widely used in clinics and animal production. We also got SDHI, succinate dehydrogenase inhibitors, QOIs, quinonoside inhibitors. They are basically targeting respiration. And then we've got multisides, like sulfur, copper, and things like that. So in terms of the mechanisms of resistance, they're basically the same, or pretty much the same ones that you will find in clinics, okay? So we've got target site modifications, the mutations in the coding sequences or insertions or deletions in promoters that leads to overexpression of fungicide targets or antifungals. We've got alternative pathways, so these organisms are mechanisms by which they can, and there's somebody trying to join, mechanisms by which these pathogens can actually detoxify molecules or just take a deep root, right? And still, just keep going with their metabolism. You've got decreased accumulation of fungicides. By, for example, the overexpression of transporters, ABCs and MFS transporters are quite common in diseases like Aspergillus fumigatus. And then you've got the overexpression of target sites. This overexpression can be of wild-type copies of the target site or mutated copies. Okay. So, starting with these Aspergillus fumigatus, so do we have the same mechanisms in clinics, animal production? Yes, we do. Right. So we've got target site modification or expression of the targets. We've got transporters, And this is not restricted to one disease, one pathogen. So this is all over the place. I've got a list here, a very short list. So for example, Candida diseases, in which, you know, these mechanisms have been identified as well. And even, you know, Cryptococcus neoformans. So these mechanisms are widespread, and they just keep repeating all over the place. Which is sometimes confusing because CYP51, which is a target site for DMI fungicides, it is actually quite conserved, but, you know, it changes, you know, it differs, you know, between diseases. So sometimes, you know, in my lab, it has happened that we had students thinking that they have found a new mutation and they were very happy. But then, you know, after you research, you know, intensively the literature, you find after some time that, you know, that mutation has been described, for example, in Aspergillus. So in order to overcome these problems, we sort of designed a tool just to simplify our lives a little bit. So we basically assign archetypes, so diseases or pathogens that are basically the template for these mutations, and then you can align your sequences there. It's just basically a blasting tool. So this allows you to identify whether a mutation that you have recently found is actually already described in the literature or not. This is just an example. So this is a little bit of an introduction to the problem, and now I wanted to target these couple of cases that are a bit more novel in terms of, you know, what the molecular mechanisms are driving resistance these days in agriculture. So I'm going to be talking about two diseases that basically impact barley production, if you like there. So these are your enemies. So you've got a spot and net forne blotch. So net forne blotch, this is a picture on the left-hand side. So with these particular symptoms, these are following the veins, this necrosis. The pathogen that causes disease is Paranophora teres teres. I'm going to abbreviate it as PTT. On the right-hand side, we've got the spot forne blotch. Pathogen causing the disease is Paranophora teres maculata. They are very similar diseases. Actually, there are some places where they don't distinguish between the two diseases. Two pathogens. We abbreviate Paranophora teres maculata as PTM. So they've got worldwide distribution. They're found in pretty much everywhere where people grow barley. They can actually be responsible for up to 30% yield losses. That's actually quite a lot. And unfortunately, there's not a lot of resistance available, you know, from the host. So we breed for yield. There's not a lot of resistance in the varieties that growers are using at the moment. So the main means of control is the use of fungicides. And you can imagine there's an intensive use of chemicals for the control of this disease. A few years ago, we found a field, some collaborators of us found a field in West Australia that was looking like this. After many treatments with DMI molecules, up to 5, we've got here tebuconazole, propiconazole, ciproconazole, epoxiconazole, and then another propiconazole, and even a QI in here. So, and after all these treatments, the crop was looking like this, not very happy. Just to give you an idea what a crop will look like after 2 treatments with DMIs, it will be something like this, a very happy and green crop. It's going to produce a lot of barley, and it's going to make the grower very happy. So we isolated PTM, Paranophora teres maculata, from this crop, or what we thought was Paranophora teres maculata. And we found different levels of sensitivity. This is over the years, right? So you've got sensitive isolates, and then what we call reduced sensitive isolates. So slightly more tolerant to the compounds, right? This is a logarithmic scale. It's a cartoonish representation of logarithmic scale, response to different DMIs. And then what we found in this field was actually this red ring, okay? That was way more resistant than everything else that we have found to date, okay? Now, we thought it was actually PTM, but the symptoms were a bit different. Actually, in the lab, we call it the leopard because it's a bit different. So actually, for reasons I'm not going to explain now, after a lot of molecular analysis, we found out that it's a hybrid. So it's a hybrid between these 2 different species, PTT and PTM. So these hybrids have been found rarely in nature, but what we found in this in this field was just this hybrid. It was nothing else, only the hybrid. It was like super comparative, and it was just displacing everything else. So these diseases have been found, these pathogens have been found to be able to undergo horizontal gene transfer. So this has been described in the literature at least once, right? So it's not very crazy to think that these two pathogens, PTT and PTM, could actually have hybridized to produce a very resistant, a very DMI resistant hybrid. So resistance can emerge in different ways. So a species at some point develops or acquires a mutation, emerges, and the resistance basically gets established in the population. Or sometimes it can come, through sexual recombination between two isolates, one sensitive and one resistant, right, to produce an offspring that is resistant. So in this case, what we're actually proposing is that two species, not two isolates, the same species, were hybridizing to produce this very resistant hybrid. So, but if that's the case, so at the genomic level, so we should see a footprint, okay? And this is basically what we found. So I've got a very summarized version of the analysis that we conducted. It was a combination of PacBio, Illumina, Nanopore. But basically, what you've got here at the bottom is our sensitive Paranophora teres. Then we've got a Paranophora teres that is resistant. At the top, we've got a Paranophora teres maculata, PTM sensitive, and then we've got our hybrid. This is at chromosome 6 that basically harbors the CYP51, which is the DMI target. Okay, as you can see, they're basically chunks of the genome that belongs to PTT and chunks of the genome that belong to PTM. So if you look a bit more in depth into that gene, you can actually see that there are particular mutations that I share, or particular SNPs that I share between one species, or they are basically incorporated from one species or the other in this hybrid resistant. But what was even more puzzling is that when we conduct the San Silico DART analysis, so pretty much everything that we found in that field was actually the same. It was a clone. So we compare it to PTM, to PDT, and even to a more distant species, barley grasses. And basically they were always just sitting close, but outside. And so they were clones, all right. Now something that we could actually understand is that this clone seemed to be moving very quickly, because originally we found it in the Esperance region, in the southeast corner of Western Australia. Then a few months later, 350 kilometers towards the west in Albany. And a year later, we found it in the north, 600 kilometers, right? So this is potentially possible for a disease that is airborne, for example, a powdery mildew or Aspergillus fumigatus, right? Clinics. But not for a disease that is stubble-borne. So stubble-borne disease moves a few meters, maybe 100 meters a year, no more than that. But this hybrid was actually moving close to 600 kilometers a year, so which is not possible. So I'm not going to explain the experiments that led to basically this discovery, but one of the actual traits that this hybrid acquired through this process of hybridization was the possibility of being transmitted in the seed. So actually what was happening is that us humans, growers, we basically were moving the seed around, and by moving the seed around, we were also moving the clone. So just a few conclusions just to finish this part. So we basically established that hybridization is a potential fungicide-resistant evolutionary mechanism. They should be actually investigated in other pathogens. Just imagine the possibility of some Candida species hybridizing to produce more resistant forms. So in addition of the chemical resistance that this hybrid actually acquired, so it also was able to acquire other traits, like for example, the possibility of being transmitted in the seed, and something that I didn't discuss here in the interest of time, increased virulence. So actually this hybrid was more virulent on varieties compared to the parents. And obviously, the the reason why this hybrid has been selected has been so successful is because of the growing systems, or the current growing systems that we've got in developed countries, where agriculture is very intensive and the selection pressure is really high. So in our lab, we also work in what we call disruptive management approaches. I'm very happy to discuss that towards the end, but it's not the focus of this talk. So the second example that I wanted to talk about was the other disease, for net blotch. Remember, the pathogen that causes this disease is Paramyxoteres teres, PTT. So here I've got two photos, the left-hand side and right-hand side, on the left. So these are treated and untreated crops. One has been treated with a combination of SDHIs, QOIs, and DMIs, pretty much a flagship of treatments that you can, at the moment, deploy in agriculture for grains. This is very expensive treatments, right? And not every grower can afford them. As you can see, there's no difference between the two fields. The right-hand side was actually treated. Just to give you a reminder again, so this is what the field looks like with just two treatments in the absence of resistance. So clearly there is a problem here. There's resistance. Now, resistance or increasing expression of the CYP51 can emerge in two ways. So one is, for example, like in aspergillus fumigators, there's 34 base pair insertion in the promoter, combined with a mutation in the coding sequence that leads to this overexpression. But there is another way. That is basically just multiplying the number of targets, basically just what we call it copy number variants, right? So one fungicide target just gets duplicated, throughout the genome. And this is a problem very often when these duplications are in tandem, right? So this is just a relationship between the target, the number of CYP51 copies in a genome, and the resistance level. And this is what you want to see. Basically, it's nearly a linear correlation, but this is what you actually see in this pathogen. So you've got very resistant isolates with low copies, okay? Same mutations, so now the difference. So the problem with this organism is it's extremely repetitive in its genome content. So, and repetitive genomes are very hard to sequence. For example, if you were to do Illumina, so many of the reads would actually collapse. If the copies are in tandem, they will also collapse in many cases. If you were to do, for example, digital PCR, which we use a lot for remutations in order to determine copy number, so you will often get numbers that are not integer because they're very close. So it's actually quite difficult. You can digest the genome and that improves, but that's not a solution. So a solution in our case was using long-read sequencing. And so nanopore is something that many of us use these days, and it's actually becoming more and more popular. So in this slide, I've got a plate with 3 different isolates, one carrying 15 copies of the CYP51, very resistant, another one carrying 6 copies, and a sensitive one carrying 2 copies, because this organism has 2 copies of the CYP51. So when we conducted this analysis, we managed to resolve this genome. So it was actually really good because we could produce, a genome with very few scaffolds. So it was actually the best at the time. But in many cases, telomere to telomere assemblies, right? But what actually really surprised us is when we compare the genomes between PTT resistant, PTT sensitive to DMIs, and PTMs. So we found that there was a break of microsynteny between the PTM and PTT species, and a microsynteny between PTT resistant and sensitive. We could actually see that in this particular example, we had 10 copies of the CYP51 towards the telomere. It was in the region with the chromosome, sorry, with the it's chromosome 6, the region where the CYP51 target is, right? And when you actually zoom in this region, you could actually see that our CYP51 copies are sandwiched between transposon elements of different types, 3 different types, okay? Now, if we know what the transposons actually do, the transposons are just there doing their thing, but when there is an environmental factor that triggers a stress, right? So these transposing elements I've been seeing in many organisms just jump, right, and land somewhere else, okay? That can actually be adjacent. They can land, just close, and that will be an explanation for the tandem repeats, or they could just land somewhere else far in the genome. Remember, genomes, DNA molecules, they are not, just a long line. They are actually quite convoluted. So they could jump, quite relatively closely, but, physically, but genetically will be actually quite distant. So in our case, these transposons have been jumping, just around, the same region. It's a 54 kb region. And the next thing is actually quite interesting because this continuous jumping actually has been modifying or what has been inducing is instability in the genome, right? So here at the top, we've got a graph just basically showing this the effect of these TEs, these transposing elements just jumping around. So this is our resistant isolate at the top, and this is at the bottom is our sensitive isolate. We're basically showing here the chromosome position. And in our resistant isolate, I was telling you that these insertions were accounting for around 54 kbs. So actually, we've been detecting deletions of 54 kbs as well, right? In other regions of the same chromosome, okay? When you have a look at the sensitive, you cannot see those deletions, obviously. Now, this is all over the place. You can see, the deletions of bigger, smaller, but, this movement of these transposing elements actually is leading to this instability. So the genome, in order to, or the organism is trying to compensate this by deleting some sections or some functions of the genome, right? The interesting thing here is that these deletions don't seem to be affecting the fitness of the pathogen or their ability to resist different stresses. Now, so what is really going on here? So in this region of the genome, what we're representing are our CYP51 copies, right? So these are the duplications. We've got 3 in this case. These green genes, these are actually effectors, right? Or effector-like proteins. So they are potentially involved in. I told you earlier that these organisms also, they acquire the capacity to infect other varieties. They could be actually involved in, higher levels of virulence, right? So you will have, in the same region, duplication of this CYP51 that leads to higher resistance to chemicals, and this duplication of effector-like genes that potentially leads, to higher virulence. It's like, the survival pack, right? And then we've got this transposon invasion, right? All over the place. You can see, that is quite important. And at the same time, the organism is just trying to block that, right? Because too much of this transposon invasion is not good, right? So there is some RIP happening as well, around 30%, right? Remember, RIP is this mechanism to inactivate transposon elements, right? And so, which is basically what we think what is happening here is that, is that some sort of a forced evolution very rapidly, because we haven't seen, these copy numbers earlier than 2017. So actually we're witnessing, this happening is now in the field, right? And the only reason probably for this, to be happening so fast is for the high selection pressure in the environment as a result of the use of the DMIs, the high use of DMIs in the field. So we go back to the original slide that I was presenting on how fungal resistance evolves. So I guess that for this, in this particular case, we have to take into account that it's not just target site mutations or insertions in promoters and things like that. So we also got genome rearrangement that is leading to a higher level of resistance in these pathogens. So I guess that, well, I don't like the word conclusions because there's still, work that is happening right now, but obviously long-read sequencing has helped us a lot in deciphering these mechanisms. So we know that these transposing elements are doing something important in the genome. They're actually changing the structure of the genome. They're breaking the microcentenate, but at the same time, they're potentially leading the duplication of the CYP51 copies in the subtelomeric regions. And probably, you know, it's something that we have to take into account in order to design management strategies going forward. And it's already been 30 minutes, so I promise I only got a few more slides, and this is just about, well, what do we do with all this, right? Because this is really nice. We publish a paper, some students get the masters and PhDs, but how do we actually translate that into meaningful messaging that can go into the field and people can use it, you know, to grow food in a safer way? Because at the end of the day, it's safer what we want to achieve. Right? So we produce tools, for example. This one here is a GIS visualization tool. I'm just showing a heat map for South Australia. This is resistance in a particular pathogen. It could be anything. It could be an insect, or it could be Aspergillus fumigators, right? But these are geo-localized samples. Obviously, this is de-identified. You cannot track farmers, but you can basically provide the industry with a up-to-date real-time map where resistance is happening and what are regions affected. Okay, on the right-hand side of this panel here, we've got management information. So we've got a big project that is basically in its almost 5th year now. It's the Australian Fungicide Resistance Extension Network. So it's all about extension of fungicide resistance within the industry. So this project has put together the only independent guide of fungicide resistance management available in Australia, and a map about basically among the few ones available worldwide. So the majority of the guides available are basically produced by the same companies that produce the chemicals. We've got a website full of resources. I've got just a screenshot here where we've got webinars, podcasts, and different basically pieces of information just to help growers with the management of this problem. And obviously, you know, we measure impact of these initiatives. This is just an example. These are agronomists that basically attended a workshop that we conducted actually here. And you can see, you know, these agronomists, you know, their response, you know, to whether they had changed, you know, their practice after attending the workshop, but they had considered, you know, changing their management practices. You can see, you know, that the majority, close to 88%, were considering that. With this, I'm just going to finish. So there's a lot of people behind this work, but mainly 2 in our group. Wesley Mayer, an extremely talented research assistant. So actually, he publishes more than a postdoc. Chala Turo, he's a postdoc, a bioinformatician, but also he's what we call a 4-wheel drive. He does, you know, bioinformatics, his witchcraft in the office, and then, you know, he goes to the lab and he tests, you know, everything that he found. A lot of organizations are contributing as well to our research, and just a little bit of advertising. If anybody's interested in a postdoc or PhD positions, we've got a few, so just email me. And with this, I'd like just to thank you for your attention. And if you've got any questions, I'm very happy to take them. Thank you.
Okay, that was amazing. I'm just going to stop the sharing. And then we'll have some questions from online first, I think.
I'm so happy that you did this.
Yeah, I know, right? It's so terrible. Okay, there we go. Sharing finished, so I'll let you stay in the hot seat. And
I could have been standing.
Oh, standing.
I was just sitting here. I couldn't actually.
Yeah.
I didn't know if I was moving or not.
So free the energy up. So questions from online participants first would be great. So I see David Guest's got his hand up. David, you go first, then Tanya.
Thanks, Fran. That was a great coverage of the issues and some of the challenges. So I've got a few other questions, but the first one I want to ask is so the movement of the pathogen in seed grain is a bit of a concern. It's also something that's very easy to manage, right? So when farmers pickle their seeds to control diseases like bunt and loose smut, don't those fungicides work against net blotch?
Some of them. So, with fungicides, we've got fungicides with a very big with very big spectrum, right? So they will be basically targeting multiple diseases. Other fungicides, they are not that good at, controlling multiple diseases, they are quite specific. So yes, sometimes these fungicides can control the diseases. The problem is that the majority of the fungicides that are used for controlling these diseases on seeds, they're actually compromised for these pathogens. So mainly there are two, DMIs and SDHIs. And actually something that I didn't say during this presentation is that SDHIs is the other group that is completely hammered by resistance in this particular pathogen.
Bring back mercury.
I used to work with mercury in the lab during my PhD. Yeah, very effective, but very scary as well.
David.
Okay, Tanya.
Well, I had one that really relates to transmission, and it's a question from ignorance. Obviously, seeds get transported, but I wondered whether some of that rapid transmission could actually be windborne.
Some of it. I'm pretty sure that if we go around these regions where we found it originally, so you will find it, you will actually see, this clone just spreading around. But it shouldn't be able to actually move 300 kilometers or 600 kilometers within the same season unless we help it, right? We couldn't, because when we didn't know it was a clone, we thought it was just independent emergent, right? So it was just, resistance was emerging independently in different locations.
Yeah.
Then when we discovered it was a clone, obviously, that wasn't possible. So we started, thinking about other possibilities. So we thought, okay, so we know that this pathogen is not moving a lot. People are moving two things. They are moving stubble, they're moving hay, not stubble, they're moving hay, but mainly actually it's coming to the east. It's not going around in the west. So the other thing that people are moving are seed, but this pathogen is not seed-borne. But, we thought, okay, let's just gonna try. Let's just gonna say, so we basically ran culture postulates. We actually saw that, if you were to infect a plant, then the seeds will have the pathogen. You disinfect the seed, you analyze the DNA in the seed, and you see the pathogen. But also, if you grow the seed again, you get symptoms in asthenic conditions, and when you isolate the pathogen from the symptoms, it's the clone. So we managed to demonstrate the postulates. So is seed-borne? That's something that we know now. So this probably, and still, we're talking, with possibilities, probabilities, right? But it's a very sound one, because the other factor that I didn't talk about during the presentation is that there is very few bulking facilities for seed. Just a few places in Western Australia, and the same happens in the East, are actually responsible for bulking the seed that then growers are going to be using. Growers hatch the seeds from their own crops, but also, buying from these bulking facilities. And these bulking facilities are actually, in places where we found the clones. So potentially people, buying the seed, they just moving the pathogen around. And the other thing I wanted to say is that it looks like, very all gloomy and this is doomed, but no, I mean, there are possibilities, there are management practices that can be deployed. It's just, there's something that we can use in agriculture very easily that we cannot do with humans. We cannot breed humans for resistance, right? We shouldn't be doing that. And we cannot use fire with, infected humans, but we can burn crops. You basically deal with the disease. You can rotate to a different crop. If you rotate to a different crop, there's no problem anymore. And you can breed for resistance. So you've got those options that unfortunately, or fortunately, I would say, you cannot use in clinics. But yeah, so there are options.
Okay, Justin, would you like to go to the next person? I do have another question, but maybe it's someone else's turn first.
Sure. And thanks for not advocating for the eugenics or incineration in human health. Yeah. Caitlin, you're the next.
Thanks so much, Fran. That was a fantastic talk. I had, several brain explosions as you were talking. Thank you so much for coming across. And I'm sorry I can't be there in person. Before I ask my question, Daniel mentioned that his video couldn't turn on, but I actually wonder, Justin, whether yours, I don't know if it's the same video, so we can see Fran talking. I don't know if that's an option.
You don't see Fran at the moment?
Oh, I thought that you were saying
No.
this.
Oh, well,
No,
we're
you
seeing ourselves, so why is that not working?
Oh, I can see Fran.
You
Maybe
can
it's in
see
a...
Fran?
Yes, so can I.
Oh.
Okay, then you must be in a different box. Sorry.
Okay, I'll work on it on my end. That's fine. One of the questions that I had as you were talking was, I just, it hadn't occurred to me how much of a crossover there would be between what's happening in the fungal, human fungal pathogen world, trying to figure out, resistance in human fungal pathogens and the environmental, and that there's a lot of stuff that we're trying to do that's already been done. And there was that website that you mentioned, Fungicide Resistance Allele Screening Tool, which sounds phenomenally useful. I tried to Google it and it didn't come up. Could you?
I'll send you a link. It's FRAST, FRAST. But I'll send you a link. Yes, absolutely. I can
That'd be
actually
great.
we can make the slides available.
Yeah.
If people want to.
Yeah, we'll
I
make
don't know what.
we'll make the slides available with all the links in the video as well. I agree, it looks like an absolutely fantastic resource.
Well, it's just, we wanted to, we came across this problem many times, and you've got very happy students thinking that they found a mutation, and they're just driving you crazy for a couple of weeks until now you realize that it's not a new mutation, it's not a new transporter, it's nothing new. It's been described in clinics. It's just that you only read papers in agriculture.
Exactly. That's happened exactly that with, Tropicalis and FKS mutations. It's the same mutation as in albicans. It's just a different annotation. It takes a while to figure that out, or it did. There are a few more papers on it now.
Along the same lines, we actually have the same problem with nomenclature. So
yes.
we put together a paper on nomenclature just to simplify the whole thing. So, and we followed the same principle. It's quite old. It's from 2015, 2014. But yeah, so because we call mutations in different ways, and sometimes it's just the same mutation. It's just that the CYP or the cytochrome B target is just a bit shorter, a bit longer, has more or less introns.
Yes.
But yeah, mutations are pretty much at the end of the day the same. Nature is just not reinventing the wheel. It's just repeating models that work.
Yeah. And I was intrigued by your approach to copy number variation, which is definitely something that's a key factor in Tropicalis and in albicans in azole resistance, and the fact that you'd use long read sequencing and you'd found that there were hybrids, and I suspect that hybrids also occur between Tropicalis and parapsilosis. So my question is, have you applied that kind of work to Candida species yet? And I can think of a few places that you could apply it.
No. Unfortunately, we just restrict our work to agriculture. I said that my background is clinics, but that's I left that behind quite some time ago. I work with we work with people in clinics. Actually, we've got a paper where we collaborated with some researchers from actually Sydney on Aspergillus fumigatus. We were providing isolates from different places. This was actually from vineyards, but we haven't done any research in Candida species. It would actually be great, collaborating with somebody, blink, where we could just help with some of this analysis, or because sometimes what I actually find very frustrating, and we have done this in our lab several times, is that you think that you're doing something new, and then you read papers in clinics, and people have done it already, for several years. So we learn from our own mistakes, and I would say that 50% of the literature that I read is actually from clinics, because the mechanisms that you said at the beginning, they're very similar, and I was saying nature is not reinventing anything. It's just repeating something that works. So yeah, we're very happy to work with somebody working with Candida species, collaborate on this. Because I'm pretty sure, that this is not a one-off mechanism. I'm pretty sure that happens, all over the place.
You're going to have your door knocked down, I fear, after this.
I have a follow-up question, but over to Tanya again or someone else.
So David and then Tanya.
I was just going to say, Caitlin, look, identifying those crossovers and commonalities in agriculture and human health is one of the great strengths of One Health approaches. And the other thing that I know we've talked about before is, look, if pyranophoras are becoming selected for insensitivity to DMIs, so are soil-borne aspergillus and all sorts of other fungi that are, you know, sometimes human pathogens. And so that means that they're being exposed to these antifungals in the field before they ever come into contact with a human, and they're already insensitive. I had another question though, Fran. So you mentioned these mechanisms of, you know, how insensitivity or resistance evolves, you know, TE factors and things like that. How much or what role do you think the fact that these fungi are haploids and often heterokaryotic, what role does that have in either driving or hindering selection for insensitivity?
Yeah, definitely it's something that we believe that, you know, it enhances the process, you know, being haploid, you know, it means that you only have to deal with one piece of information really at a time. The problem here is that these pathogens, they seem to keep always a sensitive copy. There is a sensitive copy sitting somewhere, right? And when you know, originally when we were running Illumina, we wouldn't find the sensitive copy. We only will find the resistant copy because it was in higher abundance. But then, you know, when we started, you know, doing more in-depth sequencing and more long-read sequencing, we actually found these sensitive copies sitting all over the place. It's like they reserve, you know, another card, you know, just in case they need it. This probably indicates that these pathogens or these mutated copies, they are not as effective, right? They are not very good, or they are not as good as the sensitive copies in, you know, metabolizing their substrates. And just in case, you know, these sensitive copies just sitting there. So, but yeah, going back to your question, you know, just being haploids, it simplifies the whole process. And probably, you know, just involves that the whole selection is faster. It's also a problem there. If something goes wrong, there's no going back, right? Because, you know, being an haploid organism. So that's why we believe that having this sensitive copy sitting on the side, it gives them a chance that, you know, if high and fast mutation, you know, happens, you know, in with those copies, there's always, you know, the possibility of going, you know, back to your sensitive copy and survive. Because as we know, you know, CYP51 is an essential gene.
The strengths and danger of that kind of genetic complexity is really thought-provoking. Tanya, you next, and then the last question from Caitlin.
A quick follow-up one. Towards the end of your talk, you asked what can be done with all of this fascinating information, and I think you were suggesting that modifying your fungicide use is really the key. But I wondered whether there's any gene therapy is very popular in human disease at the moment, and whether there's any potential for a genetic approach to stopping these transposons or something within the agricultural context.
Traditionally, everything that involves GM is seen as something, you know, really bad in agriculture. There is an important pushback, you know, and this is not just Australia, it's all over the place. As you can tell, I'm not from here, being in different countries, you know, doing research, and I found, you know, the same problem, or the same, I wouldn't say problem, the same situation. But probably, can I show in a slide? I've got a slide towards, or already too late.
I think it's getting too complex.
All right. Okay. No, that's all right. I can send the slide if needed. But basically, we work on what we call disruptive approaches to managing diseases, and so we use for example, I don't know, you hear about epigenetic modifiers. So we consider these epigenetic modifiers, at least some of them, as, you know, main switches, you know, that they basically control big networks of genes. So we've been working on targeting some of these epigenetic modifiers, and we have found already a few that seem to be essential in agriculture, at least, you know, in pathogens in agriculture. So we are working with them, and the idea is to develop inhibitors that actually can target these epigenetic modifiers that seem to be very specific to agricultural pathogens. The other thing we're doing is working with DNA origami. So we're working with collaborators that know way more than us in this space, and with this particular technique. So DNA origami is something that was originally developed for encapsulating viruses. So it uses the capacity of DNA to fold in particular shapes to encapsulate, particular structures. So we thought, okay, why don't we use this to, try to not to encapsulate fungi because it's too big, but just to interfere with, for example, fungal adhesion that is so important, to establish, disease, right? Fungal colonization, fungal penetration. So there are multiple structures that can actually be targeted. So we have achieved already we have basically managed to put these structures, this construct inside the fungal cells. We've got beautiful green glowing DNA origami structures inside the cells. Now the next step is to actually make them fold and target, for example, CYP51 or something else. We could target, something. It doesn't need to be essential. The key here, I don't think that we have to, achieve this total destruction anymore. So what we have to try to achieve is multiple different mechanisms operating in the same way, so that we can draw down, the effect of the fungus. By doing that, we're going to reduce a lot the possibility of one of those mechanisms developing resistance because mutations of any sort just emerge. So we've got multiple mechanisms operating at the same time. So that's the strategy. So going back to your question, genetics, can it be an answer? Definitely. So using genetics, targeting different genes that are responsible for particular processes within these pathogens is a main activity, and we're trying to achieve this, in different ways.
Okay, thank you.
Thank you.
Caitlin.
Thanks again. I thought I'd just draw you out briefly on the comment about long read versus short read sequencing. Go back to something quite basic. A lot of the human fungal pathogen work, for probably obvious reasons, is focused on short read sequencing, including elucidating copy number variation.
Yeah, and
it's cheaper.
And also because I remember when Illumina started, becoming fashionable. There was an Illumina machine where I was doing my postdoc at the John Center, and it was working, day and night. And but it was also the only thing available at the time. So a lot of the research, that was happening at the time, is still a reference. There's no reason why, we cannot just take one of those original pathogens and resequence them, right? And see what you find. Technology now is very cheap. Everybody, well, a lot of people do nanopore in their labs. We've got very, very nice and well-established nanopore pipelines in our lab. Not only we sequence whole genomes, we also do what I call amplicon nanopore. So we use UMIs just to basically, just in complex samples determine, frequencies of mutations, which is really interesting because, you're just combining the best of, for example, digital PCR platforms with, sequencing, and you're capturing everything, what you know and what you don't know, which is, very, very important to us because in many cases, you think that a particular mutation is responsible for a phenotype that you are seeing at the hospital, in the field, whatever. But actually, it's not only that phenotype, it's only that mutation. So it's that mutation combined with X number of other things that are happening at the genome level. So sequencing is an important tool these days, and thanks to long read sequencing, in-house long read sequencing, now it's very simple. Thank you.
Thanks, everybody. Thanks everyone who's joined. Thanks, obviously, mostly to Fran for such a fantastic talk. I think I've learned a lot. There's a lot of interesting potential for future collaboration as well, and just those parallels and some of the interesting approaches and the awesome resources that you shared are really exciting. So thank you again.
Sorry that it took a bit too long.
Thank you for joining us. I hope you've enjoyed today's talk from the Fungisphere webinar series, and we look forward to seeing you next time.